Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Excellent. I'm Larry. So today what I want to talk about is this problem of image captioning. So given an image, can you generate a caption describing that image? And this is one of those problems that recently has really captured the imagination both of the public and of researchers. There was a New York Times article a few months ago that probably some of you might have read. And if you think about the problem, I mean, why is it interesting? I mean, as far as for me as a researcher, what I think is cool about it is it is essentially AI complete. In order to come up with a good description for an image, you not only need to understand what's in the image, but you need to be able to understand the world in which that image exists to come up with a description which is compelling to the human. And then from the public's perspective, what's nice about it is it's something that's easily describable. I can go to my mom and I can say, and she says, Larry, what are you working on now? And I'm like, and say, describing images. And she's like, got it. So there's something nice about that. I think graduate students kind of gravitate towards that thing too, because they're like, oh, I understand that. That makes sense. I know why I'd want to work on that problem. So today I'm going to use this problem to kind of motivate a couple different things, talking about common sense, talking about vision, and language as well. So as I said, there's been a huge amount of work in this space recently. Just in the last 3-4 months, 7 papers popped up on arXiv around November timeframe. With the ICML deadline just a couple weeks ago, I think there was another 2 or 3 papers that popped up. I don't have them on the list here. But there's been a huge amount of work in this space. And just to give you an idea for this type of captions that these algorithms can generate, let me just give you a couple examples. So one I already showed you. But here's another one. A man with a colorful umbrella walking down a street. And if you look at this caption, and if you're at all familiar with computer vision, this would just blow you away. I mean, 3 to 4 years ago, you would never be able to get a realistic caption like this. And you look at this, and it's man, we recognize man, colorful umbrella, it's awesome, there's a street, he's walking down. I mean, it's indistinguishable from what a human would write. Here's another caption. A train traveling down train tracks next to a train station. Grammatically, okay, a little iffy, but not bad. And it's traveling. You might, if you look really close, you can see the bumper there. So it's probably not traveling, but it's pretty close. Again, as computer science people, we give it a break. We're like, we're impressed. We have this one. A herd of giraffes walk down the street in the middle of some trees. Again, for my mom, she'd be laughing at me. But again, as computer scientists, you're kind of like, that's not too bad. I can see the 2 giraffes. I can see the trees, the walking. There's a street. You kind of get it. Blur your vision, as Jeff Hinton would say. And of course, it wouldn't be computer vision. It wouldn't be, AI if we didn't have, a dog sitting in front of a window. The complete failure cases. Yeah. So, but it kind of works sometimes, and that's what matters. That's what has us, impressed. So, what I'm going to do is go off on a little bit of a brief tangent right now, and what I want to do is just describe one of the algorithms that recently came out, one of the ones that I'm involved in, that I find particularly interesting. I really like the model in this one. So this is work that I did with Xinlei Chen, who's a student at CMU, and he was an intern with me last spring. So let me just read a sentence for you. A girl. And I'm going to just more to the sentence. But what's interesting is when you read a sentence is that you have a tendency to, especially a descriptive sentence, visualize what the sentence is saying as you're reading it. So if you read a girl, you may picture a girl. And you say a girl and a boy. You picture a girl and a boy. And they knocked. Now you're kind of picturing a boy and a girl knocking on the door. And then if you say down the tower, then you might picture a boy and girl, knocking down the tower. And, whenever we read a book, or just, any of these kind of descriptive sentences, this happens automatically. Part of our understanding of the sentence is this being able to take that sentence in its, its form and translating it into the visual incarnation of what it's actually trying to describe, right? Now let's go and look at, kind of our simple recurrent neural network. And basically the most naive way that you would set up a network to generate a sentence from a bunch of visual features. So here you have an image, let's say, and you have in blue, it's the visual features as input. You have some sort of hidden state. I'm showing the recurrent neural network here unrolled. So you have your hidden state here is the S's. And then you have the words that you generate, which are the W's. And what happens in a network like this is the S's, or the the S hidden state basically just encodes the last few words that you stated. It's not encoding anything visual. It's not encoding a visual representation of the scene. And what happens in a network like this is it generally has kind of a short-term memory, has a tendency to repeat itself. It does kind of babble in a way that's vaguely reminiscent of the image, but it doesn't give you what you want. So what we decided to do was to build a model that looks like this. It's very similar, but we just add one layer up on top. So now we're going to take the visual features. We're going to then try to produce a sentence. But from the sentence, we're then going to try to reconstruct the visual features. And this hidden layer U, what it essentially has to do is take the words as they're coming in and try to encode the visual scene as it goes from one word to the next. And without doing anything special, like the LSTMs, the long short-term memory networks that are also popular in this space, we can actually get long-term visual memory just using a standard recurrent network. And then if we want to do sentence, use the long-term memory. So then if we just want to generate a sentence, we can chop off the top, and we have the visual features come in, and then we have the W's. And notice that the long-term visual memory is still used to help generate the sentence, because the U basically says the visual part of the scene that I've described so far. You can look at V, which is a part of the visual scene which I haven't described yet. You look at the difference of the two, and that can tell you what to describe in the future. And then if all you want to do is create a visual scene, or actually compute the visual features given a sentence, you just use the top part of the network. And you don't have to actually have the bottom. And as far as accuracy, what was surprising is that 20% of the captions that this can generate were judged to be equal to or better than human captions generated on the same images. And 20% is a pretty good number. And if you look at the other papers that I showed you, most of them are actually hovering right around the 20% too. I think another one from MSR got a bit better. I think it was like 23%. But they're all kind of around that 20% range. And like I said, that's, that kind of perked everybody up. They're like, wow, this is a task that might be kind of doable. But I don't want to talk about the successes in this talk, actually. I actually want to talk about limitations in this talk. Because I think there's been a lot of hype, a lot of hoopla around these algorithms and about how great they are. And I think that there's this perception out there a little bit that this problem is, I don't want to say solved, but we made huge progress. But what I want to talk about today is some fundamental limitations that the current pipelines that we're using have in actually dealing with this problem. So in order to talk about these limitations, let me just describe to you the space of all images along two dimensions. So one is boring to interesting. So images which are boring, images which are interesting, and then we have images which are common and images which are unusual. So you can take images which are interesting and unusual, like cats karate chopping people flying through the air. You can find images which are common and interesting. So these things that individuals find interesting, like pictures of their kids, let's say. We have images which are boring and common, like this one. Believe it or not, somebody still uploaded this to Flickr. I don't know why. I just wanted. I need to know the background story to this one, because I like, why would you bother? All right. So the question might be, okay, we have all this whole space. Where does our data set come from? Our training data set, where is that coming from? Well, let's look at the training data set that most people are using right now. So for this task, the data set that most people are using is the Microsoft COCO data set. And this is a collaboration between people at Microsoft and people all throughout academia and other industry places like Facebook. And the main purpose of this data set was to take images like these, which have a lot of objects in contextual relationships where these scenes are actually kind of confusing and realistic, and try to identify the different objects in them and segment them out. And the data set has 80 to 100 object categories, tons of images, tons of object instances. Every instance is segmented. It's a huge amount of effort. It's essentially 7 years or 8 years of somebody working full time around the clock to generate this data set, the equivalent of that on Mechanical Turk. But one thing that we did is we just happened to add 5 sentences per image. And it's that data that this community has really taken off on. So now we have, if you imagine you have 300,000 images, we have 1.5 million captions. So where do these images come from? Well, they came from Flickr. Where do Flickr images exist? Flickr images exist here. They're going to be relatively common because you have to actually be around to take the pictures. And they have to be interesting, unless you wouldn't want to bother to upload them to Flickr in the first place. Right? So let's look at the consequence of the images kind of being in that space. So let's look at another great caption generated by an algorithm. A man riding a wave on a surfboard in the water. Again, you're like, wow, this is an amazing caption. We recognize the surfboard, the man, there's a wave, he's riding it. You got to think of all the information you have to recognize in order to get this caption right. Well, let's just go back to the COCO data set and look for images which have a surfboard in them. What do we find? This image. That caption works pretty well for that image. This one, pretty well for that one too. This one, not so much. Close, the good kayakers you might mention. No, no. This one, just fine. So what you find here, when you look at the data set, you begin to question your assumption about how intelligent some of these algorithms are. Are they actually recognizing the surfboard and the wave and everything, or are they just recognizing that that is a scene which you can take this entire caption and just kind of splat it on and it'll work? Let's do another example. A giraffe standing in the grass next to a tree. Again, this is an amazing caption. You get the giraffe. You have the fact that it's standing on the grass. You had to recognize the grass, and the giraffe is standing on it, and it's next to a tree. You have to understand next. And tree, like the amount of information you need to understand seems really amazing. But let's look up the word giraffe. Okay, so the caption wouldn't go really well with that one. Here, that would work just fine. Here, that would work okay. No, no, yes, yes, yes. You see what I mean? So here again, you might just recognize when there's a giraffe, the giraffe is probably standing on the grass and it's probably next to a tree, because most giraffes are standing on the grass. So it's not as amazing as you might think. All right, I know, and then one thing I don't want to do is give you the impression that everything is just kind of gaming it in this way. So you take this caption here. Now this caption wasn't automatically generated. This is one I just made up. But you look at this image, a man walking his dog in the park. And you might ask yourself, okay, what if I look up dog? It seems like a caption that would be fairly common. How do we get, what if we look at random images with dog in them? Well, here, no, it's very cute, but it doesn't fit. No, no, no. You probably would mention the sandals. Definitely not. So it's not all categories that does this work with. And I don't think it's quite a coincidence that if you look at the good results from these papers, that there's not many dog images in them. It is more the giraffe, the surfing, that sort of thing. All right. So what we noticed is that when you have high image density, these algorithms are doing pretty well. And maybe it happens to be that 20% of the data is of this type. And that's why we're doing well in that 20%. And it's kind of in this space right here. So let's talk about why we're not doing good in the other parts of this space. What about here? What about boring and common images? So why are they hard? Well, when we were trying to create the COCO data set, one of the object categories we wanted to have was a hairdryer. The problem is we wanted 10,000 examples of these objects being used in real life in like natural situations. And it just so happens you can't get 10,000 images of people using hairdryers on the web. They don't exist. It doesn't take too much imagination to figure out why. I mean, this one, it's not too flattering. Same thing with like pumping gas. You cannot find a picture of somebody pumping gas online. It just doesn't exist. I don't know why. I mean, we pump gas every week, right? We're Americans, proper Americans. We pump gas all the time. But we never take pictures of ourselves actually pumping gas. All right. So basically, this data doesn't exist. It's too boring. Nobody wants to take pictures of these things, so we just don't have the data. It's not uploaded to Flickr, and if it doesn't exist on Flickr, we don't have it. I mean, we could have Google Glass, but it doesn't seem to be turning out as well as people would hope. So, yeah. All right. What about this other area? Unusual and interesting images. What about this one? A crazy zebra climbing a giraffe to get a better view. And you think, okay, so why doesn't this image work? Well, okay, if you're a computer vision person, you're not going to be. There exist literally 2 examples of zebras climbing on the web. This one here that you're currently looking at, another one of a zebra in a tree climbing. It's not enough training examples to actually learn what a climbing zebra actually looks like. So we can't get at that. And the other thing is, the language model is going to be screaming, please do not say zebra climbing. Climbing never, ever, 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 ever occurs after zebra. It just doesn't happen. And it's going to be screaming, just don't do it, don't do it, don't do it. It's going to force you to say something else. So our language models aren't allowing us to do it, so computer vision algorithms can't recognize it. So we're toast. Let's look at another example. A man is rescued from his truck that is hanging dangerously from the bridge. Now if you look at this image, I love this image, because if you look at it, it's a single snapshot in time, right? Yet this image is describing a whole sequence that probably evolved over a 3-hour time sequence. right? And I think everybody in this audience right now could describe what happened in those 3 hours, and our stories would be very consistent. Yet this image is just of a single particular instance, right? And that's why this image is so awesome. That's why it won all sorts of prizes when it was photographed. And if you look at the word rescued, think about the amount of information you need to understand to come up with the word rescued, even though I think everyone, again, in this audience would probably use the word rescued when describing this image. But coming up with an algorithm, a computer algorithm, which would actually know about rescue, know that this truck is about to fall, know that the driver used to be in the truck, knowing that if the truck fell, the driver would probably die, and that the truck now, the driver, even though he's hanging, in the middle of thin air, is being rescued. That takes a lot of reasoning. All right. So how can we go from here, which is where we're kind of doing good, to this entire space? And what I just mentioned here is it's a well-known problem. I'm not sure. Has anybody seen this video? If anybody interested in computer vision should watch this video. It's awesome. It's a made-up word called vimodalin, which has the made-up meaning of the frustration of photographing something amazing when thousands of identical photos already exist. I think we've all had this feeling. You get a really awesome photo, and you take a picture of it, and you take it back home, and you're like, but this looks like every other rainbow picture that I've ever seen in my life. It doesn't have this. We all want to take the same pictures. Yeah. See, I mean, you've taken that picture. I know you have. Even painters, 500 years ago or whatever, they're like the same thing. So, I mean, as far as computer vision researchers or machine learning researchers, what matters is that this is, you can think of it as the fear that our data sets will never have the diversity that we desire, right? We're never going to sample the world in the way that we want it sampled. And if we can't sample the world in the way that we want to sample it, how are we actually going to get training data? So this brings up the question, is photorealism necessary? So do we actually, when we want to learn about a world, do we need to use real images? What if we used cartoon images? What if we tried to create a world with this guy here with, let's say, a bunch of his friends, and they had a whole bunch of things that they can interact with, and all sorts of animals that can do all sorts of crazy kind of wacky things, and let's say we gave it to Mechanical Turkers, all these pieces of clip art, they could just drag and drop and create any type of scenes that they wanted to. It's just like a nice children's interface, tons of fun to play with. What do they create? They create an amazing diversity of scenes. They create really boring scenes. They create really interesting scenes. They create really funny scenes. It's just basically across the board. And this is just showing a small subset from living room scenes and dining room scenes that we are creating. We also have outdoor scenes. We have street scenes. We have all sorts of different things. And since it's fun, it's pretty easy to collect. And then we can get sentences for these scenes. So this is an older clip art data set, but if you look at the sentence, I love this. Mike trips on his way to catch a frisbee. Jenny is frustrated with his incompetence. This is an incredibly simple scene, but think about trying to come up with a sentence. Try to come up with an algorithm that realizes that Mike is pathetic at catching a frisbee because he fell down and therefore he's incompetent, and that's why Jenny's frustrated. That's a hard research problem. Another one, a cat sits anxiously in the park and stares at an unintended hot dog that somebody left on a yellow bench. I mean, what does it mean to be unattended? What does it mean to be anxious? Why is a cat anxious? Etc. It's simple visuals but complex meaning, and allows us to study this. Now, one thing you might be thinking is, but Larry, how are we going to get enough training examples? How are we going to learn what it means to be incompetent? How are we going to learn what unattended means, or anxiously? Well, that's a cool thing about clip art. You can go from a clip art, you can go to a sentence. From that sentence, you can go back to Mechanical Turk and say, generate a whole bunch of scenes which have this meaning, which have this, that this sentence depicts, like this. So let me just give you a quick example. We give the Mechanical Turkers the sentence, Mike fights off a bear by giving him a hot dog while Jenny runs away. These are different scenes that independent Mechanical Turkers made. And you can understand what it means to give your hot dog to somebody. You can understand what it means to fight off a bear. You know what it means for Jenny to run away, etc. Here's another example. I'm not showing you the sentence, and I don't want to run over on time, but I'll just give you the sentence. Jenny and Mike are both playing dangerously in the park. Right? And you look at how, the creativity that Mechanical Turkers have in playing dangerously is awesome. You can throw a baseball at a bear. You can kick a snake. You can jump off a bench. You can launch rockets off, etc. And from this data set, you can learn all sorts of interesting things. You can learn what it means to run away from. That if you're running away from something, that thing you're running away from is behind you, and then the thing that's running towards you is facing you. You can learn concepts for things that you don't necessarily think of as visual. I mean, you don't think of want as visual. But if you want something, it means that you're probably looking at it. And we can actually learn that when you want something, you look at it. We can do other interesting things. We can take the we can try to learn these concepts such as dancing with and wrestling with, or running to and running from. And it's hard to collect images which convey, let's say, running to versus running from. Right? But what we can do is we can go to Mechanical Turk and give them essentially these paper doll models, and they're allowed to kind of move their joints around in any sort of way. And basically they create this large variety of different poses. And we can learn from this and try to apply it on real images to learn what these different concepts mean. It's amazing. The creativity here is, again, never underestimate a Mechanical Turker. They just do an amazing job. The fighting scenes are priceless. I mean, I didn't want to show them. Some of them are like graphic, but they're awesome. The wrestling was bad enough. And then you can apply it to real images. And then one of the things that's important, like the rescued. When you think about rescue has a very specific temporal meaning. Rescue doesn't exist in a specific set time. It actually evolves over time. So another thing you can do with clip art is you can actually get Turkers to generate sequences of scenes, so you can learn what happens through time. So Jenny and Mike, they have a hamburger, and then a bear shows up. They both run away. The bear eats a hamburger. There's a lot of things you can learn from this. So for instance, then if you have a scene like this, and you say, which way is Jenny going to move? You can say, oh, Jenny's more likely to move forward. If bear shows up on the scene, Jenny's more likely to move backward. Good stuff. So just in conclusion, I think a lot of times, especially in computer vision, in machine learning in general, we have this tendency to kind of think of these real-world data sets and have this kind of bias towards a certain kind of style of training. And I think it's important, especially for a lot of these things, to kind of open up our minds, especially when we start going to more realistic applications, things where we want to learn common sense knowledge. Even as humans, we don't learn things firsthand. I don't know that I'll be eaten by a bear because I was eaten by a bear. Right? I learned that through secondhand knowledge. And there's a lot of ways to gain a lot of knowledge, and probably much more efficiently if we do it through other means. We do it through playing. We do it through, basically people showing us, perhaps through clip art, through these other modalities. And then from a computer vision perspective, I think it's really interesting because what's happening is as we start thinking about semantics, and we start thinking about semantics in conjunction with computer vision, we realize that we can't really think of ourselves as computer vision researchers anymore, but really need to start thinking of ourselves more as AI researchers. And I think it's a really interesting time right now to be in this space. All right. Thanks.
Questions?
Very fun talk, by the way. I'm curious to know what did your algorithm label that image of a zebra climbing a giraffe?
It's my fault. I haven't gone back to actually try, but I'm actually kind of curious as well. To be honest with you, I bet you would just say a giraffe standing on the grass next to a tree, if I had to make a guess.
There's another question back there.
Oh.
Regarding the cartoon images and learning from them, if we do a really good job in identifying a bear in a cartoon and then identifying a real bear, and if we are able to make that association, and then we have a linguistic model wherein we are able to do an association to say bear is a dangerous thing, we would still perform as well as the current research is going on, right? Like, why is it that there is a special reason so as to why we have to learn from cartoons alone as opposed to
the main both. Problem is just you can think of it almost as a pragmatic reason, which is it's easier to sample the world through cartoons and to generate this data through cartoons than it is to get the data itself from the real world. Because we can basically, and like I mentioned, we can actually go, like let's say there's a part of the world we don't know much about. We don't know much about pumping gas because nobody takes pictures of pumping gas or videos of people pumping gas. We can act that out in cartoons and get that data. Or things that are unusual, we can act that out. So I think it gives us a way of exploring the world in the way that we want to explore it without having to go and get the real data. Because getting real data is, well, it's really tough.
It's sort of like simulating the...
Yeah, totally. And it also makes a lot of the learning problems easier. Like this way, you don't have to focus on like the computer vision problems. You don't have to sit there and think, okay, what classification algorithm do I want? I know computer vision right now is doing awesome, relatively to what it was doing 3 to 4 years ago, but it still has a long way to go. And this allows us to completely bypass all those issues with computer vision not working that well and just really kind of study those higher level problems.
So, okay, one more question. Isn't it true that you're still facing, right here, still facing sort of a problem with sampling because there are so many combinations of objects, and how much can you really label?
Yes. The interesting thing there is when you start thinking, and this is something that we're just beginning to work on right now, but it's when you have active learning. Is when we're not just going out to the Mechanical Turks and say, generate 1 million scenes for us and we'll learn from it. But when you generate a small number of scenes, we learn something from it, we go out and we ask more questions, right? And you get this active learning framework. I think then that allows you to kind of explore the space in a cost-efficient manner. And they still need to kind of have a couple random samples so you can kind of discover a new space that you hadn't explored before. But it does allow you to kind of fully fill out the space, in this way. So I think that's kind of where I'm thinking that is the most compelling direction to go in for that. Because otherwise you have the same bias as the photographer bias that I mentioned before. Yeah. And also the clip art that we give people is randomized every time. And that helps a lot with creating a lot of randomness.
Great. Okay, thank you very much. Let's thank Larry. So is your talk sorry? Okay, so the next speaker is Professor Bill Noble. Bill is a professor in the Genome Sciences Department at the University of Washington. And actually, this is, given how long I've known Bill, this is the first time I've actually introduced him in a talk. And I can say firsthand that I think Bill is one of the most creative researchers I know who's working on the intersection of biology and machine learning. So I'm looking forward to hearing what he has to say. So please join me in welcoming Bill.
Thanks, Jeff. So I want to do a little proselytizing today. Obviously, given my affiliation and my title, I work on the biological end of machine learning, and I hope to tell you about some of the problems that we're working on and give you a flavor of some of the exciting directions that are going on in the field, both of genomics and proteomics. So in order to do this, I think I have to give you a little bit of background, sort of a primer on the basics of the biology we're going to talk about. So this is a picture that I grabbed from the web, but it portrays what's commonly known as the central dogma of molecular biology, which is that biological function is encoded in your DNA and carried out by proteins. So you can see DNA and proteins here. Both of these are strings drawn from discrete alphabets. So DNA is a string of length 3.1 billion bases in each of your cells, drawn from a 4-letter alphabet. Proteins are strings of amino acids at an alphabet of size 20, and they're usually about 50 to a couple thousand letters long. And one of the things that we're interested in is characterizing how these molecules encode information and get jobs done in your cell, essentially allow you and all other living things to survive. So I'm going to tell you a little bit about genomics, which is the DNA end of things, a method called semi-automated genome annotation and three-dimensional structure inference, and then also a little bit about proteomics as well. So by way of background for the genomics end of things, if you look at DNA in a cell, there are a lot of interesting questions we can ask about it. You've all presumably heard of the Human Genome Project, which was completed over a decade ago, which consisted of assaying or measuring all of the DNA bases in a single human genome. But there are a lot of other kinds of measurements that we can do about the DNA. So for example, regions of the DNA that encode for a particular gene get transcribed when they're going to produce a protein. And you can measure that transcription of that gene. You can also measure where particular proteins come and bind to the DNA in order to get various functional processes done. You can also measure sort of how the DNA is packed locally along the DNA sequence. And all of these measurements are done with a variety of different experiments, and they're generated in very large scales. So you might have, each experiment in principle generates 3.1 billion numbers along the genome, and you might have 50 or 100 or more experimental measurements per position in the genome. So there's a large consortium called ENCODE, which is NIH-funded consortium, stands for the Encyclopedia of DNA Elements, that's generated on the order of 5,000 or 6,000 of these kinds of experimental data sets. And one of the things that we've done in the context of that consortium is develop a semi-supervised, or actually unsupervised, machine learning approach to try to make sense out of this big heterogeneous collection of data. The basic idea is to try to simultaneously partition and label the genome itself on the basis of all of these different types of data in such a way that if two segments of the genome receive the same label, then they have similar characteristics in all of the associated data. As you can imagine, you can solve this using essentially a hidden Markov model, or more generally a dynamic Bayesian network, where each of the observations is one of the experimental measurements along the genome, and what is usually thought of as the time axis in this context is actually the genomic position along the genome. In practice, of course, we don't use just a simple HMM. We have to handle things like, for instance, missingness. There are a lot of positions in the genome that are more difficult to measure, and so you have a lot of missing values. And you also have to handle the expected length distributions of the various kinds of segments. But in the end, you have this unsupervised system, which you then, oh, I forgot to say, you run this system, and afterwards, it's assigned integer labels, and then a human comes along and assigns a semantic interpretation to those labels. So that's the sense in which it's a semi-automated procedure. So we can use this to do things like rediscover the fact that the genome contains genes. So what you're looking at here along the bottom are annotations of known genes in the human genome. And along the top is a representation of our labeling, where each row corresponds to one label. We've assigned something like 25 different labels to different positions here. And the key phenomenon to observe is that you have a subset of labels that are associated with sort of the beginning of genes, the middle of genes, and the end of genes, and those same patterns recur over and over again. So you can say, okay, these cluster of labels are the things that tell us where a gene is starting in the genome. Obviously, knowing about genes is not something that was new from this algorithm, but you can also use it for doing things. In addition to annotation, it's helpful for visualization, as biologists try to interpret their data with respect to all the other data available, and also identifying novel instances of elements and novel classes of functional elements in the human genome. Now, one of the things that we've been thinking about a lot recently is the fact that the DNA sequence is itself not the full story. So it turns out that every one of your cells contains 3.1 billion bases, but if you take that DNA and stretch it out, it's 2 meters long. And that is folded up inside each cell in your body. And the way in which it's folded turns out to be important for function. So this is a representation of a particular genome where each color represents a chromosome. And we know that the structure, the shape of the DNA in the nucleus affects, for example, how genes get expressed, how the proteins get translated from the DNA, and also how the DNA replicates. And we believe it's probably important for other biological processes as well. So it turns out that recently a variety of groups, including some at the University of Washington, have developed experimental methods that allow us to start measuring DNA structure in a high throughput fashion. So the basic idea is you run an experimental assay called Hi-C, and that assay outputs a matrix in which it's in principle 3.1 billion bases by 3.1 billion, although in practice we don't work at that resolution. You might have maybe 20,000 or 40,000 bases for each entry in the matrix. And the values in the matrix represent counts of how frequently in a population of cells those two DNA positions are close together in Euclidean space. Okay, so from that matrix, there is an inference problem, which is it's sort of an analog of the protein folding problem. Given all of these contacts, can we infer the three-dimensional structure of the genome? So this is the first such structure that we produced. This is the structure of the yeast genome. So there are 16 different chromosomes colored here. You can see things like, for instance, this is the nucleolus, which is a particular structure on chromosome 12. The white circle here is the clustering of a particular kind of element called a centromere. This was really the first time that we were able to visualize a full genome in this level of detail. And this kind of approach can also allow us to do things like this. This is the genome of Plasmodium, which is the parasite responsible for the most lethal form of malaria. We've measured its structure at 3 different time points during the red blood cell cycle of the parasite, and then interpolated between those 3 different conformations. And what you can see is the sort of an expansion of the nucleus and contraction as it gets ready for egress from the red blood cell. What we're hoping for, and what we've shown already to some extent, is that the processes involved in virulence of this particular parasite are structurally occurring close together in the 3-dimensional space. So there's a problem here. If you go back and think about this task that we set ourselves in the context of the ENCODE consortium, we were doing this semi-automated genome annotation where we have all of these tracks of data. And now someone comes along and says, well, I don't have a single vector from this experiment that's 3 billion bases long. I've got a matrix. And how am I going to put that into this semi-automated genome annotation procedure? So one idea is that we can use the matrix as a way to sort of encourage pairs of loci to receive the same label. So the idea is, if you're in a part of the genome where a lot of the genes are turned on, a part of the nucleus, excuse me, where a lot of the genes are turned on, and you're close to some other gene or some other place that's also in that same area, it's also going to receive the same functional label. So we've developed a procedure. This is in collaboration with Jeff Bilms, where we essentially extend the dynamic Bayesian network to use a graph-based regularization. So the idea is that if you're doing standard EM, you can formulate standard EM as doing a sort of minimizing a KL divergence between the model and the posteriors, right, the output posteriors. And then we add an additional term in which we're minimizing the KL divergence between the posteriors associated with 2 different positions in the genome. And the strength of that connection is weighted by the graph edge. So if we think these are very close, we really want their KL divergence to be small. And I'm not going to go through the details here, but you can actually show, using external data that wasn't used in the learning procedure, that when you use just our procedure, which the software is called Segway, if you do it alone with ignoring this high C data, or if you give a very strong weight to the high C data, then you do a relatively poor job of characterizing this external data, this replication timing data. But in the middle of the curve here, things get a lot better when you're using the regularization in a smart way. And then finally, one other thing that we've just published recently is an analysis of the human genome across a lot of different types of tissues on the basis of many different data sets from ENCODE, and sort of characterized the large-scale structure of the genome into different classes of what are called domains. Previously, there were a variety of different kinds of domains characterized, these quiescent domains where not much is going on, and then 2 different kinds of domains that are sort of shut down in a constitutive or facultative way, and those were already described in the literature. One of the things that we were able to pull out using this kind of analysis is a subdivision in the active portions of the genome between what we're calling specific and broad activity. So broad being, these are regions that in any kind of cell, whether it's a liver cell or a skin cell or a brain cell, those are active in the genome, versus specific, which are really specific activity for a particular tissue or cell type. So I also want to tell you a little bit about proteomics. As I mentioned, proteins are also have interesting structure. These are just images showing protein structure. But the high throughput technology that we focus on a lot in my lab anyway is called mass spectrometry. Mass spectrometry is a way to essentially characterize what proteins are present in a complex sample, and it operates not on complete proteins but on digested proteins, essentially substrings of the protein that we call peptides. Peptides go into a machine like this. This is a machine in Mike Macasa's lab in our department. And an output is a bunch of spectra. Each observation is a spectrum in which the one axis is the mass to charge ratio of the fragments, and the vertical axis is the intensity of those fragments. The canonical problem that we want to solve here, you can frame it like this. The mass spec device does a good job of getting a homogeneous population of peptides, sending it into the device and producing one spectrum. And the job of the computer is to try to figure out what was the corresponding sequence that was responsible for generating that particular observation. It's a somewhat difficult problem because the spectrum consists of two different time series that are overlaid on top of each other, essentially corresponding to suffixes and prefixes of the sequence. So in this particular picture, all these dark blue lines are ones that correspond to fragmentations, but each fragment produces two pieces, and those each make a different dark blue line here. And then, of course, as you can see, there are a lot of other gray lines there, which are maybe other peptides that are co-eluting, that are present at the same time, or other kinds of noise. So the norm in the field is to use relatively ad hoc scoring functions to do this kind of assignment. And what we've been developing is a more principled dynamic Bayesian network framework. I'm not going to tell you the details of how the model works, but the key idea is that we're trying to capture prior knowledge about how the peptides actually fragment inside the mass spectrometer, where in this case, the time domain of the dynamic Bayesian network is the mass to charge ratio of the fragments. And so that's what this model shows. Essentially what you end up doing is you say, we have a spectrum along the bottom, an observed spectrum. You have a sort of theoretical spectrum for a given peptide along the top, and then the dynamic programming of the DBN procedure essentially does a matching between those two and tries to minimize the number of insertions and deletions. And your task essentially is to choose the right peptide to generate an observed spectrum. And we've shown, as we compare this to a variety of existing methods in the literature, that at least on some data sets, this approach works very well. One of the challenges here is that the data changes pretty dramatically as different kinds of instruments are used. And so one of our hopes is that by using a learning framework, we could have our learning system automatically adjust to, say, a new platform, a new machine platform, or a new experimental approach, whereas some of the other methods may be more brittle as the technology advances. The other thing that we're working on right now is generalizing this to, any particular mass spectrometry experiment is typically generating on the order of 10 or 20 spectra per second over the course of maybe a half hour or an hour. So you get thousands and thousands of spectra generated from a single sample, maybe from a single drop of blood. And so far, the normal way to analyze this data is to treat each one of those spectra completely independently and try to figure out, okay, what generated this one and what generated this one. So we would like to instead take into account the time domain associated with this kind of experiment. So now you can think of all of the observed spectra as one big matrix, all of the theoretical spectra as another matrix, and what we're trying to infer is the abundance of those peptides over time. So this kind of joint inference you can frame as a sort of basic least squares regression. You can add in a regularization term to encourage sparsity, because we know that over the half-hour period, any given peptide probably elutes for maybe between 5 and 30 seconds. So most of the time, that peptide should not be there. And then we also know that actually when the peptide goes through the mass spec device, it has a characteristic trend, right? It should become more intense and then fade off. And so we can actually encode that in a sort of bitonic elution profile penalty, sort of like an isotonic regression, but with two directions on the isotonic penalty. And this is what we're we're currently working on. What we then want to do after that is working is to generalize this to the complete case where instead of just trying to say, here are the peptide sequences that were responsible for generating these peptides, we can also take into account the fact that we know that these protein sequences were originally broken up into peptides. And so, for instance, if you see a protein consists of 5 peptides, and we have strong evidence that these 4 peptides are there, we should be more likely to believe that the 5th one is also there. So we have now this 2 levels of many-to-many mappings from the observed spectra to the peptides to the proteins, where you have many thousands of examples at each level, and you'd like to be able to do a joint inference over all of this data at once. And of course, so for instance, my one collaborator has 5 or 7 different mass spec devices running essentially 24 hours a day. So they have tons and tons of data, and so we need to be able to do this in an efficient fashion as well, because that's just data from one machine from one half-hour or 1-hour experiment. So we've got a lot of work set out for us. There are a lot of people to acknowledge here. I've highlighted in red the names of the people who are here in the audience that can tell you more about it, and I'm happy to tell you about it as well. And I'm happy to take questions.
Time for questions. Yeah.
So I have very limited knowledge in biology, but we were learning about the double helix structure, and what you showed here today are much more complex structures of the DNA. Can you explain how these two relate to each other?
Yeah, it's a question of scale. So the double helix structure, each turn of the helix is about 10.3 or 10.5 base pairs, and then that helix itself makes another larger helix, and actually that helix then gets bound up in many other structures. So when we look at it from high-C data, we're looking at the order of 40,000 base pairs per sort of bead in the optimization. So the single helix structure is much smaller than that. There are actually lots of interesting problems about inferring local structure of DNA as well, and that's a separate problem.
So I have a question. I mean, it's actually based on what you were saying at the beginning, which is that the DNA, it's something that if you were to fully unroll sort of, and it would be a 2-meter long string, which then is bundled up together into this 3D structure, which is this tiny, tiny 3D region. And I guess, like, just, from the perspective of real-world actual objects, if I were to take a string that's 2 meters long and to bunch it up, I mean, very, very quickly it would get, very complicatedly entangled into lots of knots and all sorts of other things, which would make it very, very difficult to sort of even sequence through the string or do anything with that string. And I guess the question is, how is it possible that, especially in a DNA sequence where the actual width is so small, when you've bunch this stuff together, that it just doesn't impenetrably become entangled and inaccessible?
So that's a great question, and the short answer is we don't really fully understand how, but one thing to keep in mind is that string, well, it's actually 20 or 46 different strings, but they all have to be copied, and so there's a lot of proteins, a lot of machinery in the cell for sort of keeping track of the DNA and separating it out in such a way that you can make a complete and faithful replication of all that DNA. So that machinery is partly responsible for making sure that these things fold into some pattern that is then can be unfolded and replicated again. But it's a much more complicated story than that, and we don't know the full story.
Yeah, when you think about the mechanics, it's much more complicated than any deep model you could imagine. For example, sorry, Lee. Sorry, deep. Sorry. Wherever you are. Are there other questions?
There's one over there. Jeff, behind you over there.
Oh, sorry. You need the microphone.
Yeah, so when I was at Johns Hopkins, there was some work on synthetic genomics, so trying to create like a minimal version of the yeast genome. And I don't know if any of this would be like practical, but would there be any way to maybe like have like a hand-designed genome that gives you like more relevant data that you can train on? So something like an active learning process with that?
That's interesting. So hand-designed genome to train on, you mean for the structural inference or for which, or any of those problems? Is that what you're thinking?
Yeah, just in general, if you could get like a particular genome that you might think would provide better training data for a model.
Kind of like Larry's version of the cartoon genome.
Yeah, right. I
Subconscious
guess so.
Influence.
Right. I mean, it is true that one of the things that limits us is that we can only look at the genomes that exist in nature. And obviously, if you know, that might be one quadrant of the, presumably the interesting, not the boring part, but maybe the common part. And so getting other ones would probably be quite helpful. The question, I guess, is if they're not compatible with life, they may not be as interesting. But they could tell us what makes the difference between functional and non-functional. One thing that's happening now, so in the State of the Union address, Obama talked about this new precision medicine initiative, and the NIH had a meeting last week where they're planning to do data collection from 1 million individuals, where they'll be essentially doing this kind of genomic sequencing. So that would give us information about what the DNA sequence is anyway, but certainly having some principled way to get sort of to characterize the rest of the space, I think that would be that could be potentially quite valuable, because what we're focusing on now is characterizing the diversity within the human population, but there's plenty of outside of that as well.
Thanks.
Okay, so let's thank Bill. So the next spotlight presentation is by Reza Eghbali, and it's going to be some nice stuff, I'm sure. So.
Hi, I'm Reza. The topic of my poster is Decomposable Norm Minimization with Proximal-Gradient Homotopy Algorithm. This is joint work with my advisor, Mariam Fazal. So the problem that we consider is this norm-regularized linear least squares problem. Minimize half x minus b squared plus lambda norm of x, where the optimization variable is x, and this norm or the regularizing norm is some type of norm that promotes a specific desired structure in the solution. So lambda is a regularization parameter. The norm, the assumption that we have on the norm is that it's a decomposable norm, which if you're interested to know what a decomposable norm is, you can learn that in the poster session. The matrix A is an m by n measurement matrix, and m is less than n, meaning that the number of measurements is less than the dimension of variable x. And finally, the vector b is equal to ax naught plus z. So it's a noisy measurement from the vector x naught, and z is the added noise. So this optimization, this convex optimization problem has a lot of applications. If that norm is L1 norm, you can use that for the sparse vector recovery. You saw one example of that in the previous talk. If the norm is an L1-2 norm, that problem is good for group sparse vector recovery. And if the norm is a nuclear norm, it can be useful for low-rank matrix recovery. So a very suitable algorithm, first-order algorithm for this problem is Proximal-Gradient algorithm, which is natural for this type of composite objective function. It gives you a sublinear rate of convergence of 1 over k. it has been observed in the literature that if you do some type of homotopy continuation on parameter lambda, you start with a large lambda and you reduce it up to lambda target and apply proximal gradient. So if you mix homotopy on lambda with proximal gradient, you can speed up the algorithm a lot. So 2 years ago, Zhao and Zhang, they proved that, so they analyzed the algorithm and proved this speed up really happens. That when you have L1 norm, you get a linear rate of convergence if you mix proximal gradient with a homotopy continuation on parameter lambda under some condition on lambda target and the sampling matrix A. So we asked this question that, so that analysis was very specific to L1 norm. We asked this question that what is the biggest or largest class of norms that we can generalize this linear rate of convergence to? And the answer was decomposable norms. It's a class of norms that contain L1 norm, nuclear norm, and L1-2 norm. And so in order to do so, we derived some interesting properties for this class of norms. And we were able to show that the linear rate of convergence still holds for that class of norms. If you're interested, I'm happy to talk about the details during the poster session.